The revision video on gene mutations and gene editing, and today we're going to start off by looking at mutations. And just remember that listening to this video will not help you in terms of making sure you understand everything, so you need to do extra revision and your own reading and seek support if help is needed. So effectively, this diagram shows you gene mutations in one diagram. So effectively, if we have a look, here you see phenotype 1, which is normal. There is a mutation here. You can see that this is T rather than A, and that has caused then a change in the phenotype of the egg or sperm cell in this case, which has then taken that impact onto the offspring as a result. So a change in the genetic base sequence alters the function of a protein, and if that is in your germline cells, so your reproductive cells, that will result in passing off to the future generations. If it's just in your cells, it might cause a mutation which leads to problems in your own body but not being passed forward. Mutation is a random change in the amount of or arrangement of genetic materials in a cell. So whole chromosome mutations are of a significant importance, and they can occur during meiosis. Sorry, and that can be when you have change in chromosome sets. So instead of having a diploid cell or a haploid gamete created of half the number of chromosomes, the wrong number of chromosomes get copied across. It can be you get a change in one of the chromosomes. So for example, Down syndrome having the extra chromosome in 21, which we'll talk about in a second, or it can be a change in the actual chromosome structure. So the example of chromosomal mutations you need to know is called non-disjunction, and you'll learn more about this, or you'll be able to apply it more when you learn about meiosis. But basically during meiosis, half the chromosomes should move one way, half the chromosomes should move the other way, leading you to the normal haploid number in fertilization. However, in non-disjunction, which you can see in the red example here, one whole chromosome has moved to the one side, and therefore that egg or sperm will have an extra chromosome, and it will have that extra pair. So an example would be if that was pair 21 in Down syndrome people, because they have inherited that extra pair. What we need to focus on mainly though is gene mutations, so the mutations in individual genes. So if you remember, a gene is a length of DNA that codes for one polypeptide or for one functional RNA molecule, and a gene mutation therefore is a change in the letters, the bases. It is random, it is unpredictable, and it alters the base sequences, which then alters the sequence of amino acids, which alters the protein. It can be caused by mutagenic agents, which include toxic chemicals such as bromine compounds, ionizing radiation such as gamma rays and X-rays, and high-level radiation such as ultraviolet radiation. Mutations can occur at any time, but there is obviously chances low because, as you should know, DNA is extremely stable and resistant. There are also checkpoints in the cell cycle which try and check and remove error DNA, but obviously sometimes they get through. There is an increase in chance in DNA replication that an error might come through, and this causes obviously mutations. And there are two main types of mutations, or two main types of mutagens, I should say, from that list I just said to you. So the first type is radiation, which has enough energy to change the chemicals in DNA, and the second type is a chemical substance that can change the DNA, so maybe add extra chemicals to it, which is what tobacco smoke does. The change in the base sequence effectively then affects the primary structure, which then affects the secondary structure, etc. of DNA. So you can see here how we make up how we decide what the degenerative code is, for DNA. So if the first letter is T, the second letter is C, and the third letter is C, you would end up with serine. But if you just change that second letter from C to A, it becomes this one. So as a result, you can see how just the altering of one letter can, not always, alter. So there's a couple of different groups of mutations in genes. And the first type then is substitution. So here you can see the original sequence is GTA, and that has been substituted now in this example. Sorry, it was CTA, I don't know where I got G from, to CT, but then this G has been replaced. So that is a substitution where one amino acid is swapped for another, like a substitution in a football or a hockey game. There is deletion, which will have a much greater effect. Deletion is where one amino acid is completely deleted. So here you can see you've got G and C, you've got T and A, okay, and you have G and C there. So this one has been deleted here, so you've only got one G and C and one T and A. And again, that would have a big effect because it would cause something called a frame shift, which we'll talk about in a moment. We can also insert an extra column or an extra base pair, sorry, which would then cause again what we call a frame shift mutation. So a point mutation is one where only one triplet is affected and it's only one amino acid will change. So that's what happens when you get substitution mutations. Only one amino acid changes because that one letter has changed. So if we have a look at this one, you can see there is GAA at the bottom here and CTT at the top. That has changed to AAA. So it's only affected, this amino acid has been changed into lysine. What can happen is still a big effect. So you can have base substitutions where there's no effect. It can be what we call neutral. So this can be because of the degenerative code. So it could be that the mutation in this case has gone from UGC, just delete that because you can't see it, to UGU to UGC. Both of those code for the amino acid cysteine, so there'd be no change and it wouldn't affect you at all. You can get a nonsense mutation. Nonsense mutation is where the mutation codes changes, but instead of coding for a new amino acid, it codes for a stop codon. So wherever that mutation occurs, at that point, no more amino acids will be added on, which is a massive impact. And the last one, a missense mutation, is where it will change from one amino acid being coded to another, which can obviously have a big effect on the protein. Missense mutations are rarely positive. They can improve the function of a protein by swapping the amino acid and increase the organism's chance of survival. If this happens, this is a new allele that is likely to be passed on to the next generation, and it can cause natural selection. So this is how natural selection and evolution occurs through variation, and then it's the new allele is selected for. However, this is rare in comparison to detrimental examples. However, it is significant in evolution over time. Look at the human DNA sequence, it actually shows us a large number of substitutions in our past. These are called single nucleotide polymorphisms, and they can occur obviously in areas that we don't actually have code for, what we call non-coding regions of the DNA that don't code for amino acids. Some are associated with certain diseases, and they allow us to check if you have this single nucleotide polymorphism that you're more likely to develop a disease into the future. One disease that has a clear link to substitution is sickle cell anemia, which is when your red blood cells will actually change shape and collapse in on the cells to make a sickle cell sort of shape. And this is due to a base substitution in codon 6 of the hemoglobin protein. And that one protein causes the change to happen that is in the beta chain of hemoglobin. And basically what happens is glutamic acid is the one that should be there, which is hydrophilic. That gets substituted for valine, which is hydrophobic. And then if the oxygen levels fall, the valine molecule will then form bonds differently to how it should, and the cell will collapse, making it useless. And it can also block arteries, called sickle cell crisis. So a frame shift then is something where one change can cause a big shift in the DNA because you're removing a letter. It can be caused by deletion or insertion. So here you can see we've got CCT. but here we've introduced an extra T in there. So if we were grouping these up in threes, you could see the impact that this would have. Let's say that was one amino acid, that was another amino acid, that was another amino acid. By adding the extra letter, this amino acid is now changed. Is actually the same in this case. This one has changed, this one has changed. So it has a knock-on effect on the frame, on the rest of the frame. So deletion substitution of one or two bases, a total loss of polypeptide can occur. Every codon following the mutation changes, and it can affect everything else. If it's insertion, no frame shift will happen if it's insertion, or deletion if it's three bases exactly. But obviously, if it's just one base or two bases, you will have a frame shift, which will be a massive impact. But just because it isn't a frame shift doesn't mean it's not a radical change. So sicklecellamenia is a radical change caused by substitution. One example of a gene mutation which we are studying lots about is the BRCA gene, or BRCA1 gene, particularly, which is linked to breast cancer and also types of ovarian cancer. And basically, it affects your chances of developing breast cancer. So if you see here, specific inherited mutations increase the risk of breast cancers and ovarian cancers. Testing for these is recommended, but only when a person's individual family history suggests it. Testing is recommended in younger women newly diagnosed with breast cancer because it can influence the decisions of treatments. So you can see how much the rate or risk of breast cancer increases with these mutated genes. Basically, why this happens is because the BRCA1 gene is a tumor suppressant gene, and basically its function is actually DNA repair. It mends the double strand following interphase when DNA has been replicated, breaks and helps correct mismatches in DNA pairing. But if this gene is mutated, then this gene will no longer then be able to carry out this function. It increases the risk of other mutations because of a lack of DNA repair, and it obviously leads to increased risk of cancers, particularly in the breast and ovarian cancer, but also prostate cancers in men as well. There are over 20,000 variants of this gene, okay, so it is a huge focus point in medical science. See here, breast cancer cases increases if you have these mutations, but also other cancers as well, and it affects men and women. How random are mutations? Well, mutations are unpredictable and cannot be directed to gain an outcome. So it's difficult to work with mutations. They may occur in some sequences of bases more than others, in some parts of the DNA more than others. So introns and exons, i.e. introns being areas that we don't code for, and exons being those that do, have different mutation rates. Somatic cell mutations, so ones that are in your body only, so like your liver cells, your kidney cells will be expressed until the cell dies and then is replaced. But if it affects your testes and ovaries, your germline cells, it can then be passed on to the offspring. Genes are important, whether mutations are important, we can use something called gene knockout, and that investigates the function of genes. And we use something called open reading frames, which are genes with characteristic base sequences that we therefore can identify and study. And we use gene knockout, which is where we basically remove a gene, normally in mice, which is often used as a model organism, to see what the impacts of that are. Mice are used because they have similar genes to humans. They are mammals, but they have a shorter life cycle, so the impacts can be soon. There are ethical issues. So how does it work? Well, first of all, DNA is prepared and is inserted into the genome of the embryo. So whichever gene we're investigating, we replace one of the copies of the gene in the embryo. Remember, you get one from your mom, one from your dad. We replace one of those copies with the target gene. So, replaces the target gene with a blank copy, effectively. So we remove the gene from there. Cells where this is successfully grown, so we don't remove both genes because the mice won't survive both copies of that genes from the mom and the dad. We just remove one of them, one of the homologous pairs. And effectively, then when they're grown, they'll only have one copy, and that target gene is effectively deleted. We then allow ones that have that deleted gene to mate, and then something about approximately on average, due to chance, 25% of the offspring will have no copies of that gene, because if you've mated a male who has only one copy of that gene to pass on, and you've mated a female who only has one copy of that gene, 25% should get neither copy of the gene. Then we can look at the knockout mouse offspring and identify the traits they have to see the impacts of the deleted gene. So gene editing can be done in a natural way with something called a CRISPR sequence, and a CRISPR sequence, or the example we're going to look at is CRISPR Cas9 gene editing, and that is based on a natural system that is found in prokaryotes. It occurs at something called the CRISPR region in the genome and uses the enzyme Cas9, hence the name CRISPR Cas9 gene editing. So what is CRISPR? Well, CRISPR is a clustered, regularly interspersed, short palindromic repeat of RNA. So let's have a look at what that means. So if we look at that and break it down into steps, the repeat means the same DNA sequence occurs several times in the genome. The short bit is that it's only going to be between 23 and 47 bases in the repeat. Clustered means they generally are, the repeats are in one area of the genome only. And regularly interspersed means the repeats are separated by base sequences of similar lengths called spacers. Anything from 2 to 120 spacers are found in the CRISPR locus. So here you can see you've got a repeat sequence here with a spacer between it. Another repeat sequence, the same repeat as before, spacer between it. So that's what we're going to be talking about when we're talking about spacers and repeating units. And palindromic means each repeat sequence has a part spread the same way forward and backwards. So for example, if obviously really basic here, but if you had T, A, T, it's the same forward and backwards. So natural gene editing then, how does it really work? So let's have a look. The enzyme, don't forget, Cas9, can find specific base sequences in the genome using something called guide RNA. So it can be used by prokaryotes to find specific sequences in the genome using something called guide RNA or gRNA. gRNA is made by using transcription of one spacer and one repeat unit from the CRISPR array. So for example, if I wanted to find this bit here, I could transcribe it, and then that would attach to the Cas protein there, and then I could use that. Okay, the space, as we said, forms 17 to 20 bases at the 5' end of the gRNA, and it is complementary for the bit of DNA that we're searching for. And then don't forget there's the repeat, which will form the loop and attaches to the enzyme. So what we're saying here is this is your spacer. It's going to be complementary to the part we're looking for. So that space would be different to that space, which would be different to that spacer, and it attaches itself to the enzyme, to the Cas9, using this bit here, which is a loop sorry, I've got my word loss here, a loop sequence there. So how does natural gene editing work then effectively? Well, the Cas9 then moves along the DNA molecule, uncoiling until it reaches the base sequence complementary to its spacer. So there is the spacer, okay, and here is the complementary bit here. Once it gets there, an endonuclease cuts one sugar phosphate in each of the strands. So you can see we've got a cut here and a cut here, opening up the DNA at that point. And this break is then been used for different reasons. So it's used by prokaryotes to destroy viral DNA that has been inserted into their DNA. We can manipulate it to identify and cut out parts of DNA that we well. So here you can see different just version of that. So we take our DNA, we can cut out the unwanted sequence, and we can use it to insert new sequences if needed. So how can we actually use this then to our benefit? We can use artificial gene editing as a result. So what we can do is we can search for the required genes we just described, and we can replace it using gene editing to find faulty genes and replace them with a desired sequence. So what we can use is the adapt, basically modify the Cas9 CRISPR sequence to allow change to this sequence. So how have we adapted it? How do we change it? Well, on the 5' end of the PEG RNA that we talked about a moment ago, we leave the normal spacer, but on the 3' end, so the opposite end, we attach a primer and we attach RNA copy of the desired base sequence that we want to be inserted in. And basically what happens is the enzyme is also modified to have reverse transcriptase attached to it, so it can assemble DNA to be complementary to the RT template, the desired RNA we want to go in, and an endonucleotide is acted to cut one strand, not both. So the gene editing process put in basic steps, and this is really basic, so you're going to need to look at this and make sure you fully understand it, is as follows. If we start with this area here, number 1, the Cas9 moves along the DNA molecule searching for the target sequence identified at the 5' end of the PEG RNA. So remember, we've got the PEG RNA, we've got the 5' end which has the original spacer on it, looking for that complementary bit of DNA. At the target sequence, the endonuclease cuts one strand, giving it a 3' and a 5' end. And the two strands are separated with gRNA linked to the target sequence by base pairing. So this primer here that we want to attach then links via base pair rules. The primer binds the free end, and the reverse transcriptase attaches this primer to ensure that the DNA molecule that you want, the code you want, is inserted into the DNA. Reverse transcriptase then adds DNA nucleotides one at a time to the 3' end of the RNA molecule, adding to the 5' end as normal.